welcome back. Thanks for being here. Now, first talk in the afternoon, we're going to get Bruce Martin talking to us about state-dependent variation in continuous trait evolution. So thanks.
Thank you. Yeah, thank you to the organizers. Thank you to everyone for being here. My name is Bruce Martin. I'm currently a postdoc with my former PhD advisor, Marjorie Weber, at the University of Michigan, but I'll be transitioning to a new position at the University of Georgia soon with Jim Liebensmack. But today I'm going to talk to you about the last chapter of my dissertation and the work I did for that. Yeah, so let's get started. So something I like to say is that trait evolution is consistently inconsistent. Oh, yeah. So what I mean by that is that, right, so this is evidenced, a lot of people made this point today, but there's a really uneven distribution of phenotypic diversity across the tree of life. We have adaptive radiations, like Darwin's finches. We have cryptic species complexes, where there's been a lot of speciation, but not as much phenotypic divergence, at least to our human eyes. And then we also have these so-called living fossil lineages, where despite long amounts of time, we don't see much phenotypic change. And so, you know, there's been a lot of thought people have put into this. In general, I like to say that trait evolution is presumably affected by this vast web of interrelated and context-dependent factors. So you have some that are like more environmental, like climate or habitat, some that are more ecological in nature, such as competition, and then others you have like more intrinsic life history traits, like say reproductive strategy. And you don't have to read all those details, of course. What I'm trying to show here is that it's kind of a mess. So, right, we have like some factors that could be considered, you know, any three of those categories, really, depending on how you look at it. And then there's other things like body size, where depending on who you ask, increasing body size should increase rates of trait evolution or it should decrease it. And there's not really good consensus on that. So what I'm trying to point out here is that trait evolution processes are often more variable than any simple hypothesis can predict. And so accordingly, we need methods that embrace the inherent heterogeneity of trait evolution processes if we want to make robust and accurate inferences. So, you know, kind of getting a baseline, how do we infer which factors affect trait evolution in practice? This is a hard problem, kind of unexpectedly so, and mainly what it has to do with is that both variables that we're interested in are largely unobserved, right? You have a phylogeny, you typically have data at the tips, and you don't really know what happened during the history of that clade. So you have to infer what happened in the history of that clade, and it's not even just that simple, right? Because these two things are interconnected. One influences the other. And so this is obvious, like, right, we're trying to test if a factor affects trait evolution, so that direction of sort of information is obvious. One that's less obvious is how, in this case, if we're thinking about a discrete factor and a continuous trait, is how the continuous trait information contains data about the history of discrete states. And so I always like to have this example if we're thinking about, like, you know, how temperate and tropical habitats affect rate of flower size evolution, and I show you this clade, or this diagram with no information. What color do you think that gray clade is? Any takers? Yeah, blue. And what made you think that? The continuous traits, right? How less variable they are in those tips. So yeah, I'm just trying to show that like this isn't like a mysterious thing. Like we can pick out these things with our eyes. But what current approaches do to kind of manage this really hard problem is they use this process called stochastic character mapping or SIM mapping to break up the problem into manageable steps. And I'm sure some of you are familiar with this pipeline. You know, you start out again with this like rates of flower size evolution example. And first what you do is you ignore that continuous trait data. You just reconstruct the history of temperate tropical transitions. You map that a bunch of times to account for uncertainty. And then you repeatedly fit continuous trait evolution models conditional on those histories. And so the problem here is, I think, best pointed out if we try to kind of map how the information flows from the data to your inferences. So, right, we have our factor data on one side, we have our trait data on the other. We use that factor data to infer a factor history, and then we use that in conjunction with the trait data to infer a factor-dependent evolutionary process. And so I like to call this sequential inference because you can see that we're not getting this communication between the factor history and the factor-dependent evolutionary process. The information's only going in one direction. Really what we would ideally be after is something like this, where we have a joint model that's accounting for both of these things at the same time. And generally this is gonna give you more power, it's gonna give you more accurate parameter inferences, et cetera. So this led me to develop this new framework that I call state-dependent continuous trait evolution models. So this is based on a heavily modified pruning algorithm. I really wish I could get into the details with it. I love math. Imaginary numbers are so much more helpful than people give them credit for. So if you want to hear more about how it works, please ask me. I'm happy to talk about it. But yeah, so what it does is rather than constructing any of these sim maps or anything, it just directly calculates that joint likelihood of both your factor and trait data under a unified model, which is technically a Markov-modulated Levy process. Again, talk to me afterwards if you want more information on what that means exactly. I nicknamed this SCE or SCI, both in reference to the SSE, State-Dependent Speciation Extinction Modeling Framework that inspired it, and also because it's kind of catchy. So yeah, so this framework is really cool. It allows us to infer variation in things like the frequency and magnitude of evolutionary bursts. So right, you might have an example where you wonder if lineages and mountains are experiencing these bursts of climatic niche evolution. I'm just thinking of hypothetical examples. I'm not saying this is real. There are also long-term evolutionary trends, so something like, oh, do aquatic organisms tend to become larger over time? Put an asterisk there, because obviously that requires non-ultrametric data, so that's a big caveat there. But so far, I've been mainly focusing on how we can use this to infer variation in rates of evolution, so bringing back that flower size example that I talked about earlier. But another cool thing about this framework is that it allows us to test whether trait evolution is affected, like directly test whether trait evolution is affected by observed and or unobserved factors. And that might seem like a weird statement, so I'll break that down a little bit. I kind of like to think of this as focal and residual variation. So focal variation meaning that that's driven by some observed factor, so your hypothesis of interest, and that residual variation being the variation in the trait evolution process that's driven by everything else. And so a lot of existing methods often ignore residual variation to their own peril. And you don't have to take my word for this. There's some wonderful papers by Michael May and James Boyko that kind of document this problem in some detail. And so the nice thing about the SCI framework is that you can use it to directly infer this residual variation, right? So you don't need state data. Instead, so like in my case, when I'm focusing on rate variation, you can use that trait data to directly infer rate variation that you associate with so-called hidden states. And the nice thing is that you can do that potentially in combination with focal variation, right? You can add back in that state data and get what I like to call this triangle of awesomeness, where you have all these three things all communicating with one another. And if you're a nerd like me, that's really cool. But so I did a simulation study. That's not really the focus of this talk today, so I'm going to kind of breeze through this. But this is the basic setup. People that do stuff with SSE models, this diagram probably looks pretty familiar. So I simulated both an observed and an unobserved state, and along with a continuous trait dependent on them. And what I mainly focused on was varying the rate of transitions between those observed states, as well as the sort of the ratio of focal to residual rate variation, and seeing how that impacted inferences. And so, you know, I did kind of the standard thing. I fit various models where I constrain different parameters to be equal to one another. I calculate some AIC weights, model average parameter estimates, all that good stuff. And the bottom line that I wanted you to take away, so right on the left, we have the discrete trait evolution parameters. On the right, we have the continuous trait evolution parameters. And overall, things look pretty good. So those blobs are distributions of estimated parameter values. Those purple lines are the position of like the simulated values. And overall, we get a nice overlap. There's some nuance there. I would be glad to talk about it if you want some more information about that. But today, we're not going to focus on that. Looks pretty good. Yielded accurate parameter estimates and reliable model selection. So yeah, cool. And so I wanted to apply this to some empirical data to see how it goes with, you know, I kind of like to take it for a test drive. And so asking this question I keep bringing up, is flower evolution faster in the tropics, right? And so this is based on this intuition that in the tropics you presumably have more diverse and abundant pollinator communities, and so might you expect more frequent and rapid divergence in flower phenotypes as they try to exploit those different, you know, that greater space of potential pollinator niches. And there's also a recent paper that I thought was pretty cool where they didn't do any phylogenetic methods, but they at least showed that there was this kind of, as you'd expect, like a latitudinal gradient where you at least see more diversity in flower phenotypes in the tropics, at least in the order Ericales. So what I did was I collected GBIF and flower size data for these three clades to kind of act as my case studies. So one of them, astragalus or milk vetches, salvia or the sages, and then cicerynchium or blue-eyed grasses, which ironically aren't grasses and often have yellow flowers, but I digress. But in any case, so jumping kind of right into the results, this is sort of how I like to present the output of this model, right? Because we're jointly inferring both discrete and continuous rates and continuous traits, and sort of, at least in my case, focusing on those rates of trait evolution. So I'm painting these phylogenies with red or warmer colors being higher rates, and cooler colors being lower rates. But in any case, at least in Astragalus, there's really no support for the hypothesis. Turns out Astragalus doesn't really have a lot of tropical lineages. Lessons learned the hard way. But in any case, one of the cool things about this method, right, with being able to infer that residual variation is that you get this really cool kind of standing out of this American radiation of Astragalus called Neo-Astragalus. It exhibits some like 3 times higher rates than the rest of the clade. And this is in conjunction with apparently higher phylogenetic discordance factors in that group, which could be affecting these patterns, but also just seems to indicate in general that this group could represent a really rapid radiation. And you can kind of intuitively see this if you compare the continuous traits on, that right side of the graph to the left side at the bottom, where, in these slower rate clades, you get a lot more conserved flower, in this case, petal length phenotypes. If you do this with the old, so this is like the old SimMap method, you don't get false positives, which you might expect. As I said, there's not a lot of sort of differentiation between tropical and temperate lineages here. But you miss out on that big pattern, right? Which to me is kind of disappointing. We would like our methods, even when we don't see our hypothesis supported, to tell us, oh, is there something interesting going on in this group? So moving on to salvia or the sages, this we actually do get support for the hypothesis. So it seems like rates of flower size evolution as measured by corolla length are about 2 times higher in tropical sage lineages compared to temperate ones, with very little residual rate variation, which kind of surprised me. Again, you can see this if you kind of compare the continuous trait distributions at different parts of the tree. There might be some association. Apparently, tropical sages are known for transitioning from bee to bird pollination pretty frequently, so that might have something to do with that. And so if you do this with the old way, with the SimMap style, you probably didn't notice a difference because there is barely any difference. And that actually makes sense in this case as kind of a sanity check because, as I said, there's literal residual rate variation in this group. And so moving on to our last group, Cicerynchium, one of the interesting things here is that we really don't get any support for the hypothesis. But if you look at this, right, you might be tempted to think that some of those temperate clades I showed are evolving at higher rates. And that has to do, there just seems to be really variable rates in this group for some reason. So we get these tenfold differences in rates. And if you look at some of the tropical clades, they're kind of showing the full gamut of that rate variation. So it seems like this rate variation is kind of randomly distributed across the tree. You do kind of get a false positive with SimMaps, if you use the old SimMap-based approach, and you also grossly underestimate the rate variation. So another point for ski, hopefully. But in any case, sort of wrapping this up, this was the state of things before. What SCE models is trade this framework for a more kind of unified joint approach to inferring whether factors influence continuous trait evolution dynamics. I use this to test this idea about maybe rates of flower size evolution are higher in the tropics, and so far we're not seeing really consistent support for that. This is a talk about what we can and can't infer with phylogenetic comparative methods. So one thing I want to say, 2 caveats, is that this framework is not yet compatible with Ornstein-Uhlenbeck models and also starts to become rapidly computationally intractable as you consider multivariate traits. So those are things that I'm looking forward to in the future. I have ideas on how to tackle both of these issues, but they're complicated and success is not guaranteed. But in any case, I'll leave it at that. I'll be happy to take any questions. A lot of people that I have to thank. It really takes a village. So in any case, thank you.
And we are continuing right on with trait evolution. And this time we have Priscilla Lau, who will talk to... Oh, it went away. Will talk to us about state-dependent OU models.
Hi everyone, my name is Priscilla, and I'm a master's student in Sebastian Hörner's lab at LMU Munich in Germany. And today I'm going to talk about state-dependent OU model, which is a new implementation in RefBase, and it is used to model continuous trait adaptation. So I want to focus on the biological question that biologists may be interested in first. Our model is motivated by the macroevolutionary adaptation hypothesis, which describes how a continuous trait converges gradually and somewhat in a constrained manner to an optimum in a long geological time. And more often, the biologists will ask questions like whether or not specific phenotypic or environmental factors would affect how a continuous trait adapt. And examples include whether or not the fishes in marine habitats are evolved to a larger size than freshwater fishes, or if grazing ruminants and browsing ruminants have different optimal lengths for the tooth crown. And to model this adaptation, it was proposed by Hansen in 1997 to use the Ornstein-Uhlenbeck process, which is a stochastic, Gaussian, and also stationary process. And excuse me. And there are two terms in this differential equation. The deterministic term has the alpha as a rate of attraction parameter. And as alpha goes to 0, this term will completely diminish and it will collapse into a Brownian motion or a Witness process. And there's also an optimum parameter in the deterministic term. And for the stochastic term, there is a rate of diffusion parameter, which is the sigma here, but more often than not, we use sigma square instead. A state-dependent OU process is essentially building on top of the simple OU process, just that we have the indicated K there to represent that these sets of OU parameters are specific for the regime K. And I show here a simulated trajectory of how a state-dependent OU process might work. So you see as it switches from regime 0 to 1, the continuous trait Y will converges to optimum 1. And as it switches back to regime 0, it will try to be attracted back to the optimum 0. And before I go into our approach, I want to talk about how previously people have been doing the inference for adaptation. As Bruce has mentioned, it is a sequential, or I will call a stepwise process. First, they will infer the regime history, produce a number of maps, and then we will fit the continuous trait into the OU model on each of the maps. And in order to account for the uncertainty of the regime history, more often than not, you will use a large number, like maybe 10,000 maps. And it's very computationally intensive. And a presumably better approach would be the joint inference, which hasn't been done so far until last year, which was done by Boyko et al. And they do it under a maximum likelihood approach. But their approach has a limitation on how the regime history can be inferred, because on each branch there's maximum 1 regime shift, and it has to occur in the middle of the branch. And in our approach, first of all, is an exact Bayesian approach, and is implemented in RefBase. We will go into the details of the algorithm later. We use a data augmentation approach to create the regime history. So the locations of the regime shifts are estimated as a parameter in MCMC. So there's no problem of limitations of number of shifts there. And this data augmentation approach actually is first applied in the Muskrat model by May and Moore in 2020. And this is to show how the augmentation history can look like. And finally, our approach is highly flexible. We can specify none of the parameters to be state dependent, which is like a simple OU model. It can be fully state dependent. We can specify specific parameters to be state dependent and the other to be independent. And we can also switch one or more regimes back to the Brownian motion process and completely ignore the alpha and the theta. So going into the algorithm, we use an extended pruning algorithm, which is extended from Bjørn in 2012, which is, of course, essentially from Felsenstein in 1981, the pruning algorithm. So Bjørn has a very interesting view of how this pruning algorithm works. He treated the trait as a probability distribution. And at the tip, the trait has a mean value of the observed trait value and a variation of 0, given there is no measurement error, or etc. And throughout each sub-branch from the tip, we will calculate the updated shifted mean value due to the different alpha as well as the theta value for that regime. we'll also calculate the accumulated variance along the branch. And at the time of divergence, at the time of divergence, we'll have a probability distribution that has an updated mean and variance, and the distribution looks like this. And because we have 2 branches per internal node, then we will have 2 different probability distribution, and we will get the product of the distribution, which is like all of these are all Gaussian distributions. And we continue this process to traverse the tree in a post-order manner, and finally we will reach the root, and we will calculate the probability of observing the root of the root state given the distribution, the one single probability distribution we have at the node. And as I said, our approach is very flexible, so we can choose, the users can choose what root state they think is more appropriate. It can be the optimum of the regime. It can be an equilibrium distribution. It can also be a parameter that is estimated in the MCMC. But of course, if you want to use a parameter, make sure it is identifiable. Like in this case, it's not ultrametric, so it's probably identifiable, I would say. Anyways, so we go into the empirical testing. I use a data set of around 1,200 mammals, extant mammals, their body size and their diets, to test the hypothesis of whether or not the herbivores, omnivores, and carnivores have a different optimum value for their body size. And this graph is just to show the distribution of the extant mammals of the data we use. And we can see they somehow show a slightly different distribution, but we are not sure if the optimum is actually different. So for the models, we use a fully state-dependent OU model, 3 states, all parameters are state-dependent. And for the discrete, for the regime, the transition rate for the regimes, we also use a free Markov model, which means that the rates, transition rate in between states can change freely. And this is the result we got, the character history reconstructed through the data augmentation approach. I forgot to say, but we actually use not only placental mammals, but all kind of mammals. You can see the outgroup at around 2 o'clock. And I'm not going to go into detail into like how to interpret like whether or not the ancestral state is correct, etc. But I just want to exemplify that we are able to visualize it to the audience. And in terms of the regime shifts, in this case, the diets, we have estimated around 229 to 283 diet shifts in the whole tree. And in terms of the relative shift rates, we found that between herbivores and carnivores, it has a much significantly lower shift rates compared to all other transitions, which makes sense that like if you transit from carnivores to herbivores, probably you have to go through omnivores, in most cases, not all. And when it comes to the inference of the OU parameters, this is the alpha, the attraction rate. We can see that the herbivores has a higher attraction rate than the carnivores. Let's just focus on herbivores and carnivores for here. But alpha is not a very intuitive parameter, so we reparameterize it into the phylogenetic half-life, which means the time needed for the trait to move halfway through the optimum. And I take log because I realize it's several order of magnitude different. And we can see that the herbivores has a very sharp peak at a very low value, meaning that the phylogenetic half-life is shorter. While the carnivores has a much longer, like maybe 2 to 3 order of magnitudes more of the phylogenetic half-life. The optimum value, we can also see that the herbivores has a larger value, while the carnivores has a smaller one. So I think with these two, we can kind of conclude that herbivores evolves faster towards a larger body size than the carnivores. In terms of the rate of diffusion sigma square, we didn't see much differences between the carnivores and the herbivores. And here you may wonder, I have been skipping the omnivores for so long, and what does that mean? Like, what do these posterior means? So I have made another plot of the realized effect of adaptation, the parameter rho, which is proposed by Cornwell in 2022. Essentially, in this plot, if the distribution is towards the left, the trait is evolving more like in a Brownian motion manner, and if it is to the right, then it's more like OU. And we see sharp peaks in both herbivores and carnivores, but not in omnivores. And my interpretation of this is, at least for our character coding, it is not very clear that how this omnivores evolves. Like, the inference is not like it's not clear enough. And I suspected that, I mean, not just me who suspected that is because of the very vague definition of omnivores. In fact, like, there are a wide range, like it's a continuum of what kind of proteins they consume and also the ratio of the kind of proteins they consume. And actually, I think in last year, there is also a paper about how to categorize the omnivores, and they realized that if you are omnivores that also eat invertebrates, they have a very different body size evolution than the other omnivores. And so that's it for my presentation. In my conclusion, I want to reemphasize that we have we have implemented the first except Bayesian joint inference method to infer the macroevolutionary adaptation of continuous trait to different selective regimes. It's not officially released yet, but if once it does, feel free to test it out. And the body size evolution testing also reveals that the herbivores, mammals, evolve at a faster rate to a larger optimum, and carnivores are more in a Brownian motion manner. While omnivore mammals kind of reminds us of the importance of having very precise character coding, because after all, you use model, like when you use our model or any other model, you cannot just fit in random data without thinking what your hypothesis actually is. And so yes, that is the end of my presentation, and I think I can now put on my badge of I wrote my first presentation at Evolution. Thank you.
Our next speaker could unfortunately also not be here, but again, we have the opportunity to still hear her contribution, and I'm just going to let her speak for herself.
Hello, everyone. My name is Emily Griffith. I'm so excited to be a virtual speaker as part of this symposium today, and I'm so excited to talk to you about duck plumage color and character coding in phylogenetic comparative methods. But before I start to talk about ducks, I really want to start out by saying that I think that ancestral character estimations in phylogenetic comparative methods are really our first step in understanding trait evolution. They were my first step in evolutionary biology, where I learned how to reconstruct traits using mesquite. And now I'm teaching undergraduates how to estimate character states on phylogenies using R as their first step in evolutionary biology. And the trait I'm going to be talking to you about today is sexual dichromatism, or basically when you have members of the same species that look different based on their sex alone. And typically sexual dichromatism or other sexually related traits are influenced or go hand in hand with sexual selection, with the main hypothesis being that males are gaining bright plumage through sexual selection. So over time, you might have a trait where you have a monochromatic species in which both sexes look the same, change to a dichromatic state based on female choice or other sexual selection pressures. And this ties with the ecology of investment, basically stating that the sex that invests more in making gametes or raising offspring, typically the female, is going to want to be the limiting resource. So they are going to be dull in order to have advantages of sitting on the nest or in rearing their young. Whereas males are able to invest more energy into being bright and colorful, which make them more attractive to females and then lead to more mating opportunities. But in ducks, we actually might have an exception to this rule. So that's a female wood duck or a female mandarin duck that you see on the left there. And she is starting to have these bright orange feathers come in on the back, which is typically a very male trait. Same here with this female mallard right up here, where she's starting to have a little bit of green on her head and a little bit of more black on her rear end. And these are also male-like traits. And this is weird. Essentially, We also know that you have a dichromatic female and you remove her source of estrogen, she will molt into this male-like plumage, which contradicts this kind of hypothesis that only males have the capability to produce this bright plumage. And in fact, from when we're coding this trait throughout the clade, we see 3 states instead of 2. So instead of seeing just monochromatic and dichromatic, we're actually seeing monochromatic dull, like these American black ducks here, dichromatic, where you have one dull sex and one bright sex, and I'm going to be saying bright as male-like or conspicuous. And then finally, monochromatic bright, like these Chiloe widgeons here, where both of them have these more colorful, non-cryptic plumage in both sexes. So from a phylogenetic comparative method standpoint, what is more advantageous when we're trying to understand how this trait has actually evolved through time? Is it better to have the trait coded as these 3 states or as these 2 states, basically saying that sexes are similar or that they are different? 2 states allows us more flexibility, in part because it's more simple. A binary state allows us to do more mathematically. In this case, we would just have monochromatic changing to dichromatic at different parts on the tree. However, so far, attempts to actually reconstruct the trait this way have totally failed. Now, 3 states would then allow us to actually test more specific hypotheses. For example, we could say that if we're favoring sexual selection, we would have multiple gains. So males would gain bright plumage first through sexual selection, and then females would gain it at a later time through an unknown mechanism. Alternatively, the biological evidence suggests that females may have had the capability to produce this bright plumage all along. And thus, we might have actually started in an ancestral state where both sexes have bright plumage. Then we move to dichromatism. And finally, we have these monochromatic dull species where males lose the bright plumage based on an additional mechanism. So today I'm going to be talking about what the impact of variable character coding can have on our phylogenetic comparative methods results. And so I'm going to ask how the number of character states can impact our ancestral state reconstruction, especially at the base of the tree, and also how this impacts our evolutionary correlates. So we can use trees to also test what factors might be associated with plumage color in a phylogenetic context, and see if how we're changing the character also changes those associations. So for our character data set, I'm sure that lots of graduate students can relate to this meme where we have a novel idea and then some guy in the 1980s has also had the same idea. But in my case, Scott and Brock had a very helpful data set where they went ahead and coded all the Anseriformes species as bright or dull, monochromatic, dichromatic, and then also had variables related to our potential hypotheses as to what traits might be correlated to these different states of plumage color. For our tree, the duck tree has been historically really difficult to resolve. That's in part because we have a lot of hybridization and introgression happening over a very short period of time. But in birds, we're actually fairly spoiled in that we have a resource called birdtree.org where we can go ahead and input a list of species, and it's going to come out with a simple tree based on a limited set of the genome. So I went ahead and downloaded 10,000 trees from this website. Credit to Yetz et al. For doing this work, and then summarize that tree with Tree Annotator, and actually was able to get some pretty good resolution on the different main clades in the tree based on physical and behavioral characteristics. So for evolutionary correlates, I mentioned earlier that we have some hypotheses related to why ducks might have different coloration through time, and that's our main question. So some of the variables we're looking at are if they're migratory or non-migratory, so if they're staying in the same environment year long, or if they're moving, and also if that environment is mostly tropical or mostly temperate. For our behavioral variables, aggressive behaviors, more specifically territoriality, might be an impact on why males might have bright plumage. And then parental care, more specifically male parental care, ties back to that ecology of investment that I had mentioned earlier. So for our results, in using a two-state coding, so this is just monochromatic and dichromatic, we do have associations with male parental care and migration. More specifically, that dichromatic species are more likely to be migratory and have males that provide limited parental care. Over here by our sea duck clade, you can see this orange for the dichromatic species, that mid-green for no care in males, and then that light blue showing that they are regular migrants. This also is repeated in the diving clade and then sporadically kind of around the dabbling clade. Now for our three states, it's really interesting because once we move on to being monochromatic bright, dichromatic, and monochromatic dull, we actually gain latitude as another positively associated variable. So in this case, dichromatic species are more likely to be in more northern latitudes. So going back to our sea duck clade over here, we can see dichromatic species are more positively associated with these light pink color, showing that they're in more northern latitudes. Same over here with our diving. And again, across the dabbling clade, we see that association as well. So more importantly, I think a main takeaway from this is that we do see a gain in a significant relationship by changing these 2- and 3-state character coding, which is insight that we otherwise wouldn't have if we had just stuck with the traditional 2-state coding for this trait. Now, for our reconstruction, I went ahead and started with our 2-state model. The equal rates model was best, so forward and backward transitions were equivalent. As you can see on the tree, we have a lot of what seems to be moving back and forth between these yellow and oranges, kind of across. But our main question about what is happening at the base of the tree is still totally unanswered. It's entirely ambiguous whether or not the common ancestor in this group was more likely to be monochromatic or dichromatic. Now, for our 3-state character reconstruction, the all rates different model was best, although the ordered states model did come in a close second, where dichromatism was the intermediate state, and doing so produced very similar results. Again, at the base of the tree, though, we are totally ambiguous as to what was happening here. And so I was looking at the data and I noticed something that was weird with the source that I was using. Specifically up here in this clade, the whistling clade. So whistling ducks are a group of brown ducks. They are, in terms of colors, not very colorful, but I'm going to argue that they actually share a lot of the same traits that we see in other bright males. So here are some comparisons. We have a male northern pintail here, which is a male in a dichromatic species that has these very long and, like, I just want to say elegant plumes basically on its side. And then we also have this plumed whistling duck here that also has just these huge feathers that stick out like a sore thumb. I really encourage you to look them up. The pictures are kind of crazy. Same with this male green-winged teal here. This is called vermiculation, this fine barring that we have here. And then on this whistling duck, we have that same trait, along with just a lot of contrast, which certainly doesn't lend itself well to essentially blending in. And whistling ducks are a monochromatic species. So what would happen if we code them as monochromatic bright? Well, if we change essentially the 4 kind of most ostentatious birds, we start to see that at the base of our tree, monochromatic bright gains a little bit more influence. If we change all of them, all 8 out of over 100 plus species, monochromatic bright suddenly has much more influence as to what we think that the state is at the base of this tree. So how do we code objectively? Like, this is an issue if we can just code, a handful of characters out of the whole tree, and suddenly this really impacts our reconstruction. And I'm going to say that there is a very limited way to do so. I think that we can define our traits as best that we can, but there's always going to be some subjectivity in how we code. However, I'm going to say that we can supplement this by using genomics. More specifically, we can use phylogenetic comparative methods to essentially compare the evolution of a trait to the evolution of the genes behind them. Our colleagues, Lavretsky and all, did this with DD-RAD data and mallards and closely related monochromatic species, and looking for peaks of differentiation, and found one here on the Z chromosome that is very closely associated with an area where TYRP1 is a known gene associated melanin production. And if we identify the genes related to traits and then reconstruct their histories, we're also able to evaluate the evolutionary processes of these trees. So for example, for duck plumage, we can understand if the multiple transitions that we're having are really homoplasy, if they're introgression, or if they might be the result of shared ancestral variation. So to conclude, I want to start by saying again that I really think ancestral character state reconstructions are our first step in understanding trait evolution and are a great entry point for evolutionary biology. But before we even start doing these ancestral character state reconstructions, we have to prioritize how we code the characters, because we all, I think, should acknowledge that we have biases in coding these characters. But I would encourage people to try variable coding, basically like how I used 2 and 3 state in this study. It might provide novel insights, allow us to test new hypotheses, or unveil new evolutionary correlates. And finally, I would encourage people to use phylogenetic comparative methods to inform genomic studies, because then we can also understand the evolutionary processes behind the traits we're trying to reconstruct. I'd like to thank our collaborators and funding sources for this project, and also thank you for your attention and the organizers today for allowing me to present virtually, since I couldn't be there for medical reasons. If you have any questions, feel free to email me or reach out to me on social media. Thank you.
Okay, everyone, it's almost approaching coffee o'clock again, and for the last part of this session, we have Sophie-Luise Heidig, which will take us deep into proteins and marine cyanobacteria.
Okay. Hi, everyone. I'm Sophie, as mentioned before. I'm doing my PhD at both free universities in Brussels and Belgium, supervised by Jean-François Flo and Wim Branken. And I would like to take you a bit on a detour into the space of proteins at the example of marine Pseudococcus cyanobacteria. Those are one of the most ubiquitous cyanobacteria we have. They're one of the primary producers in the ocean, and they're globally distributed, and therefore they experience lots of different environments characterized by different pHs, salinity, and of course temperature. These different environments cause them to be under a constant pressure to evolve on a molecular level. The environment affects all levels of the cell metabolism, and if you are only one cell, that's really important. So some of these effects are transient, like expression of heat shock proteins, and others get permanently recorded is mutations in the genome sequence. Especially for protein sequences, the proteins being like the workhorses of the cell, simply exchanging one amino acid to another can render the protein unfunctional. But they are also famous for being able to tolerate completely drastic changes in amino acids. Like there's families of proteins that share only 10% of their amino acids, yet they perform the same task. They look the same, they have the same shape. And it's a really fascinating space to be in, especially if you use sequence-based methods to infer their evolution. Proteins can tolerate drastic sequence changes as long as their 3D shape, their biophysical interactions and behaviors are conserved. The protein fulfills its function, and we wouldn't expect any effect on the organism level. So these biophysical behaviors form a constraint on evolution that is not really captured by standard methods, I would argue. And now I would like to quickly introduce you to the data set I'm working on. It's a very nice database from people done in Roscoff in France that has 54 highly curated, well-annotated, nearly complete genomes that provide me with a data set where I can run comparisons between ortholog proteins on a genome level. And since I am first in my PhD interested in temperature adaptations, I picked the heat shock protein as an example to guide you through this talk. HSP20 is a regulatory element in the heat shock response of Synechococcus, and it is structurally and functionally highly conserved across the tree of life. You also have HSP20s, and they are like a weak chaperone in humans. They're more important for Synechococcus. Yes, and the first point that I would like to make here is you can have a very stark difference between how you would reconstruct the evolution of a species versus if you're only looking at a singular protein. So both of these trees you can see here are created in a similar manner using IQ tree with very solid bootstrap supports. The tree on your right, the species tree is created using like informative elements of housekeeping genes that have been concatenated, while the other tree is created only from the alignment of my protein, HSP20. And you can see that there's some colorful lines in between that indicate how this tree gets rearranged depending on which part of the equation you want to look at it from. And a bit more clearly, I've made it here. You have these two sister clades, golden and red. They are very closely related the species are closely related, but the proteins seem to not share this relationship as much. There's an inverse example as well. So there is more to protein evolution than what we can capture in the sequence. So the sequence, we think, holds all the evolutionary information, but there are some interesting properties to proteins that are not directly obvious from the sequence. One of these examples would be protein folding. Up until very few years ago, it has been thought as a mathematically impossible problem to solve, to predict from purely the sequence a three-dimensional structure. However, that has been obliterated with machine learning models. The most popular one being AlphaFold. I hope you have heard from it. If not, you should check it out. It's really fun. Yes, but they also come with their own sets of limitations. The first and foremost being AlphaFold uses multiple sequence alignments to create these three-dimensional structures. So if you run it on a set of closely related sequences, you will get the same result. It's a bit of a circular logic. Another interesting constraint is that AlphaFold predicts only the final state of folding, but not the steps in between. It also has a very static view of the protein, but proteins are dynamic entities. So protein interactions are mostly guided within itself and also with other proteins by transient biophysical interactions. So a more probabilistic view of a protein at a per residue resolution is likely more informative for the purposes of investigating evolution. And as I just made the case that these three-dimensional informations are very informative for proteins, I have created a pipeline how to create multiple sequence alignments of protein sequences informed by their three-dimensional structure. I presented the poster on this early in the conference. If you were there, hi. If you're curious about this, I'd love to talk about it more after the session. It's published, it's on GitHub. If you work on proteins in some capacity, you might want to check it out. But the main point that I want to make with this slide is that while sequence variation can be very strong, the structure will be retained within a protein family, and therefore it's more interesting to align your sequences based on these structural and structural elements versus the concordance of it being the same amino acid. And now that we have established this, we can look into some very interesting transient interactions. So for example, one of my supervisors groups, the Bio2Byte group, we have the Bio2Byte tools that are a suite of different predictors that, with some fancy math that I don't quite understand, can predict from sequences probabilistic views on different characteristics. For example, the dynamics of a protein or propensities to form a beta sheet. A beta sheet is a certain way an amino acid can fold, where it kind of flips in on itself. And that's a very stable interaction, so it's very interesting if you're doing protein design, but also just for proteins to function in general. So an interesting point that I want to make with this slide is that you can see there is way more than 8 peaks in this plot. However, we know from the crystal structure and from the little view down there that there's only 7 beta sheets that can be seen in a crystal structure. So imagine like a freeze frame. So something's off here. And I noticed this very interesting peak in the middle that I could correlate with a potentially transient beta sheet that is only formed if the functional dimer is formed. This orange version of the HSP20 structure is not from Zynecococcus, but it's from a plant. So they are sequence-wise, they are not at all the same, but structurally, as I mentioned before, it is conserved. That's why I wanted to investigate this a bit more. So in not just looking at a singular sequence, but looking at all my sequences together, you can see that this specific region has a quite low coverage. That is because it is a loop that I pointed out before. The length of these loops in proteins can vary quite a lot. They are known to be of different lengths, like as a general characteristic. So it's not entirely unexpected, but it's still interesting. And it allows me to still move on with the core idea of my PhD, which is using ortholog proteins to disentangle the influence of common ancestry, which you can see represented here by a cute little phylogenetic tree that I made of HSP20, and the environment. So if I take all of these sequences and cluster them only by the temperature at their isolation site, that being the mean temperature over 10 years, which is the best measure that I could come up with. Oh, you lost a little bit of resolution here. The arrow points to a small peak in the distribution of very high temperature adapted proteins. So like you have to imagine it now. You can't see it. But it kind of correlates with the story that I've been telling to you before, which is that there is encoded in the sequence in this specific region and a higher probability than you would generally expect to form a beta sheet. And that can be useful to stabilize a transient interaction, like meeting another protein that you need to be connected to perform a function. And this is some very preliminary data on my first attempts to apply phylogenetic independent contrast. And this is a plot of the correlation of the temperature versus the per residue beta sheet propensities that I just showed you before. So to kind of show, or in question in my data, if this peak in distribution from the high temperature adapted HSP20 sequences can be mainly correlated to them sharing a common ancestor, or this being a result of a temperature adaptation. And with a moderate amount of correlation, I can at least infer that I am on the right track, and I'm very interested in exploring this more in the future. With this, I would like to thank you guys for your attention, for sticking around, my 3 supervisors and colleagues, and leave you with my take-home messages, which is 1, structure is really informative if you create multiple sequence alignments and do investigations in the phylogeny, and biophysical behaviors should be included if you're working on proteins, if you want to gain a better understanding on how they evolve. And I'm happy to take questions if you have any. Yes.
I have a hazy memory that in Synechococcus genomes, this is when there was less data, there was a great range of GC content. So, and so much so that you saw a, variation in amino acid usage. So on the one hand, is that, for example, have implications for the correctness of your trees? And on the other hand, do you think there's some heat adjustment there that's associated with underlying GC content?
So the question is if the influence of a GC skew in the general genome is influencing this analysis. In Synechococcus. Yes, in Synechococcus in particular. I have not noticed an influence in this GC skew, but I also haven't specifically looked for it. But my methodology, apart from the tree, where I'm not sure if the influence would be significant, but I will look it up. That's really interesting. The rest of the methodology should not be envisioned by that because it works on amino acid basis. So once you have the amino acids, I think the GC skew isn't. No?
Some amino acids, the same amino acid can be encoded by GC-rich codons or AC-rich codons. Sometimes you see.
Yeah.
It does actually in these species have an impact on the amino acid usage.
Yeah. I will look it up
So,
then.
yeah, I thought it was, given your interest in proteins, maybe there's something interesting there.
In the proteins. Okay.
Yeah.
I will look it up. That's really interesting. Thank you.
Great, I'll make a start. So hi everybody, I'm Bethany. I'm currently a postdoc in Tanja Stadler's lab at ETH Zurich. But today I'm going to be presenting to you a project that Tanja has graciously allowed me to do on the side, led by Will Gertie, Pedro Godoy, and Ali Chiarenza have also been lending a hand. And we're particularly looking at what fossils can and cannot help us to infer about continuous trait evolution. So today I want to talk about a specific PCM pipeline. Some of you in this room may have used it yourselves. So it involves building a phylogeny or taking a phylogeny from the literature. And then for each of those tips in that tree, we want to collect continuous trait data. So typically this might be something like body size information or some sort of anatomical measurement, but increasingly this could include something like latitude or some sort of measurement of an animal or plant's niche, or even some sort of biochemical measurement. And then in order to infer how this trait has evolved over time, we might fit a series of different models to this phylogeny and these trait data. And then we might use some sort of process to evaluate how well these different models fit and use this to make some sort of assumption about how this trait has evolved through time. So many of the papers in which this process has been applied have made kind of piecemeal commentary about how well they feel it has worked. But in addition to this, there's also been a handful of studies that have looked very specifically in a more systematic fashion at how well we think this actually works. So the first one is shown here, and this was by Slater and colleagues who demonstrated that including fossils in your phylogenies when you're trying to infer how this trait evolution has happened over time can be incredibly helpful, and they also present a method where you can add this information about fossil taxa which actually are not included as tips in the tree as well. In addition to this, there's also a paper led by Cooper and colleagues looking specifically at Ornstein-Uhlenbeck models. So in this study, they found that particularly for small trees that are ultrametric, it is quite often the case that an Ornstein-Uhlenbeck model fits the best, even when something that is more simple has been used to actually simulate the trait. But then recently there was also a comment on this paper that suggested kind of two major findings. One, that it's not just about OU. Actually, maybe this is something more general that needs to be explored in a broader fashion. And also, a lot of this comes down to how we actually interpret the results. If we're just looking at what the best fit model is, maybe the findings that we have will be more short-sighted in comparison with what we would find if we were actually looking at things like the parameters that we're inferring in these models. So kind of to summarize all of this, we felt that it's clear that there's a lot that we don't know about how this analytical pipeline works. And so we wanted to add to this by specifically addressing 3 questions. The first is, we feel that it's good to include fossils in our phylogenies, but does the age of these fossils matter? The second is this effect consistent, dependent upon the model that we're using to fit to our traits? And then the final question is, do fossils also affect how well we can infer the parameters in these models that we're fitting? And so in order to do this, we simulated a range of phylogenies, which I will talk about in more detail on the next slide. We then simulated continuous trait evolution using Brownian motion, Ornstein-Uhlenbeck, and also accelerating and decelerating modes of evolution. So for all of these different models, we used both a weak and a strong strength. And also for Brownian motion and OU, we also used models where the root state and the mean and the present were both the same and different. We then inferred the best fitting trait evolution model for each of these traits that we simulated, and we did both of these things using the R package NVMorph. And then we compared the best fitting model and parameters to those that we had used to actually simulate the trait. So in total, this looks like quite a small amount of work, but actually ended up in nearly 1 million different simulations. So there's a lot to summarize. I apologize for some very busy graphs that I'm about to show you. So I mentioned telling you some more about the phylogenies that we used. So we simulated phylogenies with a range of different sizes, ranging from 50, the smallest, up to 1,000 tips. We also included different proportions of fossils in these trees, ranging from 0, so ultrametric trees, up to trees where 95% of the tips were fossils. And as I mentioned, we were also interested in the ages of these fossils and how important that was. And so we used 2 different diversification rates to simulate the phylogenies. And we also used a subsampling process in order to create phylogenies that either had fossils which were particularly old, so towards the root, or fossils that were particularly young, so quite close to the tips, and then a random mix in between. All right, so that's the kind of brief overview of what we did. So now I'm going to start taking you through some results. So I will guide you through what these graphs are going to show you. So you can see, hopefully on the x-axis, that what we have is the proportion of fossils in the tree, ranging from no fossils on the left up to 95% fossils on the right. There is also different line types depending on those 3 different distributions of fossils in the tree that I mentioned. And then hopefully with the different colors, you can see the different sizes of phylogenies that we used as well. And on the y-axis, what you're seeing is the proportion of simulations in which the best fitting model fitted that with which we simulated the information. So in this example, what you can see is when we simulated under a weak Brownian motion model, how often were we getting Brownian motion as the best fitting model? About 75% of the time, which doesn't sound super high perhaps, but there's a little bit more going on here. So something that's really interesting, in fact, about this specific graph is that actually we see the highest success rates when we have no fossils in the tree, which is something that we perhaps weren't expecting, and opens this door to the fact that actually this issue is much more complicated than we initially thought. So the next thing I want to show you is I just spoke about when we're using Brownian motion without a trend in the mean value over time. When we do have this mean trend over time, the first thing which is immediately obvious here, hopefully, is that when you have no fossils in your tree, you cannot infer a trend in that mean value through time. This is quite commonly known and is also included in the guidance if you read it in NVMorph. So it's good that we kind of reiterated that. But then hopefully something that you can see as well is that when we have an increasing proportion of fossils in the tree, most of these lines that are lowest, that are recovering this model as the best fitting are mostly dashed lines. So these simulations that are performing poorly are the ones where we have recent fossils. So it seems like if we want to do a good job of inferring that this trend is there, then we need to include fossils towards the root that are old in order to do that. So next I'll move on to the results for the Ornstein-Uhlenbeck models. So on the left-hand side, you can see these are the simulations where we used a weak Ornstein-Uhlenbeck model. And hopefully you can see quite quickly that generally it is very, very hard to fit an Ornstein-Uhlenbeck model when the simulating effect is very weak. However, if we then look at the strong Ornstein-Uhlenbeck, again, you can see this effect where when we have no fossils in the tree, it's quite hard to fit this correctly. But then hopefully you can also see that we actually hit this plateau very quickly. So when only 10% of the tips in the phylogeny are fossils, we very quickly increase the proportion of simulations in which we're fitting the correct model. So we only need to put a few fossils in our tree in order to correctly fit an OU. And then moving on to accelerating and decelerating trait evolution. For both of these models, when it's weak, there's kind of a very variable ability for us to fit the correct model. But most of the lower lines in this case are, again, when our fossils are recent rather than towards the root. When the effect is strong, we see very, very different results between these two models. So first we'll talk about the strongly accelerating evolution at the top. So you can see we have this big range in how successfully we can fit this model correctly. And hopefully you can also see we have this gradation by color. So what this is showing you actually is maybe not what you might initially expect, which is that the bigger your phylogeny, the less likely you are to successfully fit an accelerating model. So actually, as the phylogeny gets bigger, it becomes more likely that instead you fit an OU model in this case, which is something that we found really interesting. But then on the flip side, when we're looking at this strongly decelerating rate, which is also sometimes called early burst, and I think is something that a lot of people would like to see in their data sets, actually we have some really good news, which is that pretty much all of the time we actually have quite a strong success rate in correctly fitting a DC model. So I also mentioned looking at the success of parameter estimation. So here you can see our estimates of the strength of the Brownian motion at the top for weak Brownian motion and at the bottom with stronger. So we have black lines corresponding to the correct value. So regardless of the kind of simulation setup, all of our estimates are roughly around the correct value with which we simulated, which is a pretty good sign. The precision is actually better for weak Brownian motion, which is quite interesting. And actually, we don't really see that big of effect based on how old the fossils are, but there is more of an effect depending on how big the phylogeny is, and that seems to be the biggest kind of controlling factor. And then the last thing I wanted to show you today is the estimation of the strength of a trend when you have trended Brownian motion. So again, you can see that when we have 0 fossils in the phylogeny, you're always going to infer that there's 0 trend. But then as soon as you add any fossils at all, we are starting to see that the estimate of the strength of the trend is kind of centered around the strength that we're using to simulate. What we can see, though, is that when the fossils in the phylogeny are young, we're seeing much more noise around that value. And when we're instead using fossils that are older or fossils that are evenly distributed across the tree, we're seeing much more success in being able to estimate the strength of that trend. So to kind of wrap up, I also wanted to say that all of this comes with a caveat, which is that in our simulations, we assumed that the phylogeny is inferred correctly, both in terms of topology and branch lengths. We inferred that the tip states were correct, and also this probably means that we're inferring that the ancestral states are pretty solid as well. And so this is probably not the case in a lot of empirical applications, which is only going to add more noise to what we're trying to show you today. And so to conclude, some major points that I've mentioned are that phylogeny size remains a highly influential factor in trying to fit the correct model for continuous trait evolution. Sometimes fossils help in the case that the true model is Ornstein-Uhlenbeck or accelerating or decelerating, but actually with Brownian motion, it seems like this may not be the case. It seems that often the first few fossils that are added result in the biggest gains. So even though the fossil record is sparse, just adding that little bit of information could be incredibly useful. And also estimating the strength of the trend, the strength of a trend requires old fossils. And this study was just pre-printed on Friday, so if any of this has interested you, then please go ahead and take a look. And I'd love to take your questions if you have any. Thank you. Yes.
Did you look at tree shape? In other words, balanced trees versus, cones, unbalanced tree category?
So the question was, did we look at tree shape? The answer is no. So because we wanted to balance having kind of two discrete diversification rates, but then also being able to sample fossils within those phylogenies, means that we're probably skewing the trees in some ways that we didn't quantify or didn't fully anticipate. But that is something that is very interesting and probably important and would make a great follow-on study from this. Thank you.
All right. So now we're going to hear from the last member of the Brazilian crew in the symposium, Bruno do Rosario Petrucci.
Hey everyone. Yeah. Can I start? I don't. Oh, okay. Hey everyone. Thank you for coming, and thank you for Orlando and Mariana for, the honor of being here. I'm Bruno. I am at the Heath Lab at Iowa State University. Oh, okay. I'm at the Heath Lab at Iowa State University, and I'm really interested in all things extinction rates. Today, I'm going to be talking about a case study I did with Canidae about state-dependent diversification rates with fossils. So, I'll get into that. Why Canidae? Well, when we're trying to study these complex patterns throughout the tree of life, it was really nice to have a group with great extent diversity, right, represented here by our gray wolves and red fox, and my personal favorite, the Maine wolf, back in Brazil. Well-sampled fossil record, for example, with Borophagus and this lanky boy, Hesperus ion, and even some recent extinction with, for which we have ancient DNA, like, for example, the Falkland Island wolf. So it's a really well-studied group, really charismatic, so there's a lot of, we understand, and it's really interesting to study these complex questions. So what question are we looking at today? Well, what I'm really interested in is how does diet affect extinction rates in Canidae? They have a large diversity of dietary strategies, at least, as far as carnivory is concerned, right? You see hyper, meso, and hypocarnivores well distributed throughout the tree, both in extant and extinct groups. We have this kind of preconception that maybe extreme specializations like hypercarnivory might be an evolutionary dead end, right? You increase fitness by quite a bit by being able to, being so specialized and so good at that specific thing. There's a lot of reasons that hypercarnivory, we expect, increases speciation rates, but that might also come with increased risk and extinction due to a lot of irreversible changes possibly and loss in adaptability. In 2020, a paper by Believing is also talked the other day, so I hope you watched her talk, showed that this pattern might happen maybe at the clade level, but at the species level, extinction rates didn't seem to be increased, at least for large hypercarnivores specifically. But we really want to test this same pattern with model-based methods to see the extent of these patterns. So the specific class of models we're going to be focusing on are called state-dependent speciation and extinction, or SSE models. The diagram here, we have the simplest one, BISSI, binary SSE. We have a trait evolving throughout the tree between 0 and 1. You can think of that as hypercarnivorous or non-hypercarnivorous, for example. And then species take on speciation and extinction rates, or lambda and mu, dependent on which trait they have at a given time. We can make this a little bit more complicated with something called HISSI, or hidden SSE, where now we also have a binary hidden trait. So we're just allowing for the idea that not all traits of interest, not all traits that might be affecting the diversification of our group are included in our data set, right? HISSI is a really important model specifically because it alleviates a little bit of a constant problem in SSE models. It's kind of a false positive issue where the models tend to infer that a neutral trait has effects on diversification rates, even when they're neutral. So that's something to keep in mind and a reason to not focus specifically on just one model at a time. Now, if we're talking about estimating extinction rates specifically, we show in a paper currently in review through simulations that fossil data really are necessary. So here we have posterior mean estimates for across 100 simulations of extinction rate, posterior mean estimates of extinction rate. In two scenarios, in green, we have mu0 greater than mu1, and in orange, mu0 equal to mu1. And we see that when fossil sampling is 0, that's represented right here, we have our posterior mean estimates centered quite far from the true values represented by these vertical lines, right? So our accuracy is pretty low. When we do include fossils, even with fossil sampling as small as 0.1, we see a much better accuracy of our extinction rates, estimates, and passing this idea that, again, which is a frequently talked about idea in macroevolution, that we really might need fossils if we are asking questions about extinction rates. Now, if we want to include fossils in our tree, first we need to estimate that tree, right? The most used model, or model class at this point, for including fossils in our phylogenetic tree are the FBD class of models, where we're simply taking a normal birth-death model and including a fossil sampling parameter psi. We can think of it as generating a tree something like this, right? We have our extant species and our fossil specimens distributed around the tree. I colored it just to remind you that we also have a trait evolving through it, right, eventually. But specifically for canid data, there's a lot of issues with using traditional FBD model to estimate that tree. The fossil record in Canidae is very taxonomically structured, and FBD is a taxonomic independent model. There's a lot of fragmentary fossils, like a tooth, that taxonomists are pretty confident on assigning to a species, but we can't quite say, for example, that they have the same exact morphological vector as another fossil or as the extant member of that species. So there's some challenge in how to structure and how to match that data to the model. That's not really a problem with the data or the model, but in general with how they match together. Furthermore, this is not an issue specifically with FBD, but if we're trying to kind of force a bifurcating tree into these data, we're going to have some issues because for canids, and I assume for other groups, we have a lot of situations where we have sister lineages, and one of them might be much older. We know through the fossil record that they have originated way before their other lineage. So here is just an example, right? We expect this lineage to be older just from the fossil record. And if we force a bifurcation, we force that speciation event here, we kind of create a ghost lineage, right? Where this second lineage probably appeared a bit after, and we are biasing our divergence rates through this, right? Our divergence time estimates. Now, the model that I'll use instead is called stratigraphic range FBD. Now we're using data as data, not specifically individual fossil specimens, but stratigraphic ranges, right? Dates of first and last appearances for each of our species. Now we are attaching morphological and molecular data to a species, right, to a range, as opposed to individual specimens, which again is much more in line with how paleontological data, especially canids, but paleontological data in general, is structured, right? You can think of how we mostly use morphology to assign species in paleontological data. So obviously this model is a little bit more in line with how our data is usually structured. Furthermore, it does generate a budding tree, which again means that it's going to solve both of the problems we mentioned with one tick, right? We ran this model on Beast2 using the S-ranges package, and we obtained this cute little budding tree. I don't know how many of you have seen a budding tree before. I'm really excited about that. We have our colors representing our fossil ranges here. And I'll just do a little quick tangent to tell you just one nice thing that I totally didn't just need my canid specialist to explain to me 5,000 times to finally understand how this is really interesting on our budding tree. We have here Xenocyon, a extinct canid, really big body, really big head, hypercarnivore, and they are kind of direct descendant of Lycaon pictus, the African wild wolf, one of the cutest canids, and that's scientifically proven. They have a much smaller body, but still bigger head, right? And there's this sort of narrative where you might need to get really big, right, especially on the head because of the specific adaptations required to become hypercarnivore. But then after that happens, you can kind of slim down the body, get a little bit more agile and fast, right? Now, our budding tree kind of provides another data point for that narrative, where we have Epicyon here representing a larger body hypercarnivore, and Borophagus as the smaller body one, again, still pretty big-headed. And our budding tree kind of directly implies this narrative because the Borophagus clade is coming directly from the Epicyon lineage, right? Obviously, with bifurcating tree, you could ask these same sort of questions, but it's interesting to see how budding can kind of directly provide us with interesting insights just from looking at the tree, right? Now let's take a step back real quick. What was our guiding question? How does diet affect extinction in Canidae? What do we need to apply SSE models, right, to a tree? So after all this time telling you how cool this budding tree is, I'm gonna bifurcate it. Fun, right? So yeah, unfortunately we can't quite integrate SSE models with those budding trees, the stratigraphic range model. That's something to look forward to. So we bifurcated this tree, we separated those fossil ranges into fossil specimens, and then we applied BISI to it with RevBase. So let's look at those results real quick. We have here posterior mean distributions for speciation and extinction rate estimate, rates, I guess. And we see one thing that we expected, right? Speciation rates for the hypercarnivores, that's state 0 here, are much higher than for non-hypercarnivores. But what we didn't expect, not even are extinction rates not the same, right? They're not higher. They're even much lower for hypercarnivores. And this is the kind of thing that should give you pause when you're looking at SSE models, right? You're saying, okay, this is a surprising result. It would be really cool. I'll just send it to Nature and be super famous. But, you know, let's take a step back and think, okay, maybe there's some more nuance going on, and this surprising result comes from my method, right? Not quite the data. So now let's look at the results for our hidden state SSE model. So here I have the graph specifically for hidden state B. A and B in this case are kind of arbitrary. We don't know what the hidden trait is, but if we take specifically just for hidden state B, we see the exact same pattern, right? Higher speciation and lower extinction for hypercarnivores. Now what happens if we look at the other hidden state A? We see a completely, almost opposite pattern, right? We have lower, speciation rates for our hypercarnivores. We have a much higher extinction rate for our hypercarnivores, right? So it seems like there's specifically some other trait working in concert with hypercarnivory to generate these extinction rates. And that only became clear to us when we applied HISI, right? When we allowed the possibility of not having all of the information in our model, yes, what I said, not having all of the information that we needed on our data, right? So I think this is a really interesting result to present at this symposium, right? What can we not know with these current PCM methods? That's kind of ATM machine, isn't it? Sorry. Is that we have the situation where you can explore under the hood and understand a much more nuanced idea of what's happening when you allow for the possibility that you don't have all of the information, right? And I think, yeah, this is a really interesting application of these SSE models. Oh, I forgot to pass you this one. This is just all of them together, no new information, but I think it's cool to see all of the rates together. So, take a step back, right? What are our conclusions? Budding trees can be really interesting when you're asking macroevolutionary questions, right? Getting those insights, even just looking at the tree, I was very excited. There is some signal pointing to an effect of diet on diversification rates, but it's a little more complicated, right? There seems to be another trait working in concert with diet specifically, and that's something that we really need to explore to understand these patterns fully. So obviously, on my future work slide, the first thing, include more traits, right? Body size is an obvious and easy one, probably gonna be the first thing we try. That original 2020 paper I mentioned looked specifically at large hypercarnivores, right? So it's an easy fruit to pick. Develop a more involved workflow, right? To understand, kind of parse out the effect of these different traits and the possibility of some false positives here. If you were here 2 sessions ago and last session, one of those, and watched Laura Alencar's talk. They have a really interesting workflow. Go read that paper if you haven't. We are excited to learn from them. And then we would really like to kind of develop these methods further, right? Be able to or implement an SR-FBD model that includes both budding and bifurcation, integrate that with FSV models, really expand the toolkit we have to explore these patterns. And with that, just want to thank my co-authors and collaborators and funding, and see if anyone has any questions.
Thanks. Well, Bruno brought us via the fossils back to diversification, and now we're leaving the fossils, but we're sticking with diversification because Jérémy Andréoletti will be guiding us through the congruent diversification histories that we're also scared of.
Hello everyone. My name is Jérémy. I'm a second-year PhD student in Paris with Hélène Morlon, and today we'll be talking about non-identifiability, something I would say many people have been scared about for the past 5 years. And in particular, we will apply that to diversification histories, but I hope that this talk will be of interest to most people doing statistical inference. So in our case, what we're interested in is reconstructing this diversification history of organisms with lineages that appear and go extinct over time. For a long time, people have been using the fossil record to reconstruct this history based on paleontological data. But in the past, say 2 decades, people have been developing more and more methods to use phylogenies of extant species based on genetic sequences to infer these rates in the past. And some results that have been discussed a few sessions earlier. But yeah, 5 years ago, Luca and Penel published a kind of bombshell article saying that we will always have, for this kind of model, an infinite number of rate trajectories over time that will be impossible to distinguish. So this is the problem of non-identifiability we'll talk about. And so many people have been asking this question: should we stop inferring diversification histories from phylogenies of extant species at all? So yeah. So my goal here is to first try to explain this in more detail, this question of non-identifiability, then look at some of the solutions that have been proposed in the years following this article, and including the one I'm interested in, and look at an empirical application at the end. So let's start with a global framework of statistical inference. So we never have access to the real world, to reality. We only have access to some kind of data that is produced by this reality that we can observe. And we have in mind a set of models. And all we can do is to compare these models to the data we have and basically compute what we call the likelihood, which is the probability of this data for each of the model. And then hopefully find the model that best explain what you observe. First, you can have something that we call practical non-identifiability. It's when the likelihoods are too close to clearly distinguish some of these models. But this is a practical problem, and hopefully if you keep gathering more data, at some point you will manage to distinguish your models. But in some cases, you have what we can call theoretical non-identifiability or asymptotic non-identifiability. And here the problem is much deeper, because in some cases, you can demonstrate that whatever amount of data you manage to gather, some models will be completely impossible to distinguish because they will have the exact same likelihood. So this is the problem everyone is frightened about. In our case, again, reality is a diversification process. What we observe is these phylogenies of extant species. Then we have a series of birth death models with various rate trajectories over time, so speciation and extinction. And in this case, what Luca and Penel showed is that you can have this kind of diversification histories that look very different, but are part of the same congruence class, which is defined as the set of scenarios that has the exact same likelihood. So this is a problem. And what people have been proposing since this paper. So the first thing you can do is to use simpler models. So for example, in the field, people have been using piecewise constant models for a long time, skyline models. And Legrid and Fairhost showed that these models are identifiable. And even more generally, all the piecewise polynomial models are identifiable. So if you gather enough data, at some point you will converge to a single trajectory, which is good, but I'm a bit hesitant about this solution because, okay, you will converge to a single trajectory, but you have no way of telling whether or not this is close to the true continuous process generating your phylogeny. So be careful. The next solution is to use fossils. If you can use that in a way, that's my favorite method. And it has been demonstrated that this birth-death model is identifiable when you have sample ancestors, so fossils that give rise to other lineages. So yeah, by the way, I have a poster if you want to learn more about various types of fossils with the model with heterogeneous rates. I'm waiting for you at the next station. But now we'll focus on the two last solutions. So the first one is to use what we call generally regularization. So the idea here is pretty typical in science. When you have several hypotheses that you cannot distinguish with your data, you will want to privilege the ones that are more parsimonious, the simplest explanation that manage to explain your data. So hopefully this will reduce the congruence class to a smaller set of hypotheses that are closer to the true process. And so in Bayesian inference, this will mean having priors. And something that is important to understand is that Bayesian inference is always identifiable, in the sense that if you start from well-defined priors, you will obtain well-defined posteriors. Maybe very wide and dependent on your priors, but this is well-defined. And the last one is to just embrace the uncertainty and explore the congruence class. So this has been made possible by this package by Sebastian Höhner, Bjørn, and Drew. Two of them are here in the room. So this is a really cool package that allows you, when you have inferred a rate trajectory over time, to explore alternative trajectories in the congruence class. So for example, you can sample alternative extinction rates and use this formula to go backwards in time and reconstruct the corresponding speciation rates in the congruence class. Or you can do with more complex rates, distributions of functions. And you can do the other way around, generate alternative speciation rates and get the corresponding extinction. And you can see one on the first limitation is that here you get negative extinction rates, which we know to be, just doesn't make sense. So this paper was so good that I wanted to improve it. So what we did is to look at some limitation of this. So first, you get negative extinction rates, but the other main limitation is that you cannot cover a bit more complex scenarios. So for example, if you have a declining speciation and spike of extinction, you have no way with this method to cover this kind of scenario. And so what we are proposing is to introduce something that we call the congruence class. So the idea is that when you are reconstructing these trajectories backward in time, you can take any position on this space with speciation and extinction rates. And if you want to remain within the congruence class, you have to be on this curve, which corresponds basically to the formula I showed you before. What was available in crabs was to do it in either one direction, from speciation to extinction, or the other way around. And basically what we are proposing is to do it as a joint sampling. So you're basically sampling on the congruence class, and you construct directly trajectories both for speciation and extinction without to choose one direction. And so another advantage of that is that you can constrain the rates to remain always positive, so you avoid this part of the state space where you have negative extinction rates. And so what we do is to sample between lambda equals lambda I minus 1, so constant speciation, and lambda I equals lambda I star, which correspond to constant extinction rates. And so what you obtain with that? So you have this position between those two parameter values over time. And so you get a range of trajectories from very flat extinction rates to trajectories with a lot of signal on extinction. You see that you can never have constant speciation, because otherwise it will get you negative extinction. So this is, from this constraint, you can extract a lot of the signal that is in the congruence class. You can even give more flexibility with these horseshoe Markov random field trajectories. So what you want to do now is, okay, you can generate thousands of trajectories using this, but then you also want to select the most parsimonious one. Remember this regularization proposal. So the idea here is to compute smoothness scores with, you can try various penalties that give similar results, and then use the various thresholds to keep only the one that are the most parsimonious. So we applied that on a big mammal phylogeny, inferred rates with ref base. Then you can take the median estimated rate to construct a congruence class, and then look at all the congruent trajectories that you can obtain with this expanded Krabs version package. And so if you take a very high threshold, we'll get a lot of trajectories that are a bit crazy. And if you restrict them to only the most regular one, you will converge actually to something that is quite close to the initial rates inferred with ref base, which seem to point toward the idea that autocorrelated rate priors that I use in ref base manage to find parsimonious trajectories within the congruence class. Then I want to finish by thanking Helen for supervising this project and all the other researchers who gave us feedback on this. And some of the key points, non-identifiability, problem with birth-death models. You can simplify these models. You can use fossils, which is really good. You can regularize, and Bayesian inference is always identifiable. And you can explore the congruence class, especially if you're doing maximum likelihood. I'll let you read this magnificent quote. Thanks. So, question? Yeah. Hi. Thanks.
Is there, you mentioned that like the parsimonious criteria that you chose is like smoothness of the, is there a particular rationale like behind that? So is there like why having a smooth lambda trajectory, for instance, is more parsimonious than a really variable one?
I mean, I don't know, with the way it's implemented, you can really easily change the kind of smoothness criteria. So if you want to try others, but yeah, I think basically the reasoning is that if you can explain the same data with rates that are either smooth or going all the way up and down, you want to prefer the simpler explanation because there's no reason why this rate will evolve a lot if you can explain it with a simpler method. Yeah. You have time for another one? I spoke much too quickly, fast. Okay, so. Oh, maybe sorry. Yeah.
Ask for more smoothness in the diversification, the speciation rate, and less smoothness in the extinction rate?
Yeah, I think basically what I did in the case of mammals was to sum the smoothness for speciation and extinction, but you can easily change the formula to have various smoothness criteria for one or the other. So, absolutely. Sophia?
I was just wondering about the Bayesian inference that you talked about. You said it's kind of like identifiable if you have priors. It depends on constraint or reason prior, right?
Yeah. I mean, if you're talking about practical identifiability, it's not always the case because if it's really flat, you can say like, okay, it's not really identifiable. But in the theoretical sense, given that the posterior is always well defined, even with very flat priors, in this sense, it's always identifiable. Because, for example, in maximum likelihood, if you have an infinite number of trajectories that have the same likelihood, the maximum is not even defined correctly. So that's the main difference between these two. Thanks.
And we are heading for the last talk in the symposium. Thanks for sticking with us all day. It's been quite an amazing ride. And to finish it off, we're actually going to crank up the speed a bit more because Bjørn Kopperud is going to show us how to infer diversification rate classes super fast.
Thank you so much. Should I start now?
Well, theoretically, we have to wait a little bit.
Okay, I think I'll just begin. So, well, Yeah, I didn't prepare any jokes, sadly. we should have all been better prepared with that. So maybe someone else would be more suitable for that. There we go. Ding ding. Okay, hello everyone. My name is Bjørn Kopperud, and I'm a PhD student supervised by Sebastian Höhner in Munich, Germany. And today I want to talk about how we estimate diversification rates on phylogenetic trees. So just to set the stage a bit, one thing that you might notice if you study evolutionary biology is that sometimes there can be very stark differences in species richness if you study clades that are sister taxa or of similar age. So here I've got two examples. If you know the ginkgo tree is represented by only one species, whereas its sister clade, the cycads, are represented by 280 species. And similarly, we have the lungfishes and the coelacanths, which are represented by 8 species, and its sister clade, the tetrapods, are much more successful at 30,000 species, extant species. So one question that I had as part of my work is that this is might be very obvious in textbook examples, but my question is this phenomenon something that's unique or just to these clades, or do we see this stark differences across the tree of life as a whole? So in order to answer that, I am using a phylogenetic perspective, and I'm using the birth death model, which has parameters that control how often speciation events happen and how often extinction events happen. And if you use the standard birth death model, then the assumption is that these rates are equal across all of the branches in the phylogeny. And to answer the question, we would like to ask or see if we would like to tell if some clades in the tree branched at a different tempo than others. So, and turns out that it is possible, and we made a package for this. It's called PESTO. It's an acronym, and it stands for Phylogenetic Estimation of Shifts in the Tempo of Origination. And this, if you run PESTO, you might get a result like this. So I plotted the primates tree, and you can see on this figure that the speciation rate changed on this branch, which led to the clade of the old world monkeys, and the speciation rate rose by about 50%. So we implemented this PESTO package in the Julia programming language, which I heard some mentions of already. So, and I really like it. So I would like to advertise it a bit as well. It's as fast as C, but it's easy as R in Python. So if someone is curious about this, I would recommend checking it out. I also have the link there, but I will show it later on. So if you want such an analysis, you might ask, why did we make another method for this? Why didn't we use one of the more established ones, like BAM or the birth death shift model, multi-type birth death model, or CLAD-S? But there are also others. The thing about these methods that exist is that they all use some kind of simulation approach for the inference. Be it Markov chain Monte Carlo, they might use stochastic character mapping or data augmentation. And the problem with this is that it can be a bit computationally slow. So what if my tree looks like this? I have 20,000 taxa, and I do not necessarily want to wait for a really long time. I don't want to wait months for MCMT to finish, and even if it does finish, it might not converge, or I want to use different priors. So it's a question of patience. So let's talk a bit more about PESTO. So PESTO is equivalent to what we call the birth death shift model, which is implemented in RevBase. And here I have a scatter plot showing branch-specific speciation rates using two methods implemented in RevBase and in PESTO. And if you use the equivalent priors and so on, each dot corresponds to one branch in the tree. Then the two methods are identical if they lie on the diagonal. So the top right cloud represents the old world monkeys clade, and the rest of the dots represent the rest of the phylogeny. So the two approaches are in fact give the same result. The big difference lies in the runtime. So if you want to use RevBase, you can do this on a modest sized tree. Let's say you have 200-300 taxa, you might be able to run this analysis in a few days in RevBase. But if you have 20,000 taxa, then you might have to wait 4 or 6 months. Maybe it's possible, but I'm not so patient. So instead, I would recommend to use pesto instead, which finishes on the order of seconds or minutes or maybe a few hours if your tree is exceptionally large. So you might ask, how do we do this? We don't use MCMC. What's the trick? Basically, we use what's called the backwards-forwards algorithm, which I will explain shortly here. So the first step is the backwards pass, which I highlighted with green arrows. So we start at the tips, and we calculate the probability density of observing the tree. And then we do this in a post-ordered iteration of the tree. So we first visit the tips, then the internal nodes, and then the root. Now, when we finish step one, we do something similar. We still compute probability densities, but in the reverse order. So we start at the root, go towards the more recent nodes, and we end at the tips. And it turns out this is sufficient for calculating the posterior state probability of being in any rate category at any time on any branch. And once we know these state probabilities, we can calculate the mean speciation rate, for example, for each branch, which is essentially some kind of weighted average among our rate categories and weighted average across the branch time. And what's also nice with pesto is that we can calculate the number of rate shifts. It's a bit more complicated with the differential equation, but we can use numerical methods to solve it, and it's relatively quick and easy. So main selling point is no MCMC whatsoever. Just the deterministic algorithm. So here I will show the primates tree again. And the really nice thing about pesto is you can also estimate where the shifts happen on the tree. So here I've colored the tree as a heat map, where more red means the number of shifts. We can see how big the diversification rate shift was, and we can also calculate the significance test of this shift. So each dot in the scatter plots represents one branch in the tree. And for our significance level in the space vector calculation, we had one branch that had a strong statistical support for that there was a rate shift. So what this more efficient methods allows us to do is to really broaden our taxonomic scope. And therefore, we apply this method on much bigger phylogenies than we saw here. This is about 200 species. But we applied it on much bigger and more species-rich phylogenies, and therefore I have summary figures instead of a heat map. So these are all empirical phylogenies. On the left column, I have aquatic animals. On the second one, I have terrestrial animals. Then I have flowering plants and ferns, and mushroom-forming fungi and lichen-forming fungi. So let me just walk you through the axis. So on the y-axis, it's a bit cut off, I suppose, but it's on the y-axis, it's the number of diversification rate shifts, and these are broken down. These are the distribution as a function of how large the shift size is. So the closer this is to 0, this means that there is a minor shift, but there are many shifts that are a smaller effect on the diversification rate. But there are also some less frequent shifts, more to the right or more to the left of 0, that have a larger impact on the diversification rate process. So if we take, for example, the rayfin fishes, you can see that this is a bit of skewed distribution to the right of 0, meaning that the rayfin fishes experienced an overall acceleration in the tempo of diversification. And if we look at the mammals, they're mostly balanced. So it's equal amount of upshifts and downshifts. And if we look at the bird phylogeny, there was a overall deceleration in the tempo of diversification. But one main takeaway here is that for all the phylogenies that we studied, we never found any phylogeny that did not have any rate variation. So it's pervasive in all of the empirical phylogenies that we studied. But back to the acceleration versus deceleration topic. It's possible to, instead of looking at this as histograms, we can summarize this using what we call the magnitude. So it's essentially measuring the same thing. If you have a positive magnitude, that means an acceleration, or negative magnitude, that means a deceleration. And there is a relatively balanced number of phylogenies showing acceleration versus decelerating pattern, so no strong signal there. And it's also not time related. So even for young phylogenies and old phylogenies, we saw that there was equal frequency of whether they showed an acceleration or overall deceleration. Another summary, a way of summarizing this was we were interested in the rate shifts themselves. How often do shifts in the diversification rate happen? And this is, we can explain this with the shift rate. And one really strong pattern that we found is that if we see on the left side of this graph, these are young phylogenies. For these, the diversification rate shifts happen much more often, about an order, 2 orders of magnitude more often than for old clades. So this is a very striking time scaling pattern that we saw. With this, I just want to summarize, take home. We developed this new fast method that can be applied for really large trees that have exceptional species diversity. And it's not just more possible, but it's also feasible now. You can apply this for a 30,000 species taxa. You can go get a coffee or a snack, and then you can go back, and it's finished. So no more waiting 6 months. Yeah. And this really allows us to apply this model for a much more broad scope of phylogenies, and therefore we can also learn much more about the process of diversification, which is great. And we really found that there was among lineage rate heterogeneity in all of this phylogenies that we studied. So with that, I want to thank everyone for listening, and thank my supervisor, Sebastian Höhner, and everyone in our lab in Germany. And if there's time for questions, I could take one or two.